Hello dear students, welcome in this channel Patel Science Academy. Today's topic is pollen-pistil interaction. What is pollen-pistil interaction? It is the interaction of the pollen grain, this one which is the male part of the flower, with the sporophytic tissue of the stigma, this part. So it is the interaction between the male and the female part of the flower, that is the pollen grain, and the sporophytic tissue of the stigma. So this interaction in between these two pollen and stigma is called pollen pistil interaction. It begins with the process of the pollination. As you know, the pollination process is nothing but the process of transfer of the pollen grain on the stigma of the gynoecium, and this process is called pollination, and it marks the beginning of the pollen pistil interaction, and it ends on the fertilization process, the process of fusion of male gamete with the female gamete. So fertilization pollen pistil interaction conclude, and it begins on the pollination. All the events from the deposition of the pollen grains on the stigma to the entry of the pollen tube into the ovule by the synergy method are referred as the pollen pistil interaction. It includes all the events from the deposition of the pollen grain, means when the pollination starts, deposition of the pollen grains on the stigma takes place, and after that the development of the pollen tube uh, occur there. So all the events from the deposition of the pollen grains and the formation of the pollen tube, formation of the male gamete, and its entry into this nucellus, all these events includes the pollen pistil interaction. Pollination does not guarantee Pollination does not guarantee the transfer of the right type of the pollen. Often wrong type also land on the stigma. So it means pollination is not 100% perfect process for the pollination, for the transfer of the right type of pollen grains on the stigma. It means some unwanted or wrong type of pollen grain can also be carried by the process of the pollination on the stigma. So pollination does not guarantee the transfer of the right type of the pollen grain, means always pollination is not successful because it may also carried the wrong type of pollen grains. So it goes for the pollen pistil interaction first, which is very specific process. So the pistil has the ability to recognize and accept the right or the compatible pollen on the same species. This is very specific process. Pistil has the ability to recognize and accept. This pistil is very specific, and it has ability to which one is its own species pollen grain. If a wrong pollen grain has been transferred, it doesn't recognize it, it rejects it. That's why it is called pollen pistil interaction. Thus, the wrong type of the pollen is discarded or rejected or cancelled by the pistil. It means it depends on the pistil nature. It is having very important and specific ability to recognize the its own species of pollen grain. Compatibility and incompatibility of the pollen pistil is determined by the special protein. So this compatibility and incompatibility in the pollen pistil interaction is decided by or determined by the protein, special protein. This process involves the pollen recognition followed by the promotion and the inhibition of the pollen. Means after the recognition, after pistil has accepted the pollen grain, it goes for the promotion and or the inhibition of the pollen. The stigmatic surface of the flower refuse other wrong type or the incompatible pollen grains. So means unnecessary or unwanted or wrong or different species pollen grains has been discarded or rejected because it is wrong, it is not from the same species. A physiological mechanism operates to ensure that only intraspecific pollen germinates successfully. So it indicates that the intra means the same species pollen germination successfully takes place on the pistil. The compatible pollen absorb water and nutrients from the surface of the stigma and germinates and produces the pollen tube. Means if the same species pollen grain has been transferred, it start absorbing water and nutrients from the surface of the stigma and start germinating, developing and produce the next step, that is pollen tube likewise. So you can see diagrammatically here pollen grain has transport on the stigma. If it is compatible, if it is correct or right pollen grain, it will be accepted by the stigma, and this pollen grain will start absorbing water from it and nutrients from it and will produce a pollen tube, and likewise this process will go on. Its growth through the style is determined by the specific chemicals. Now this pollen tube, it goes through this style and start further process of development, which will leads to the formation of the zygote. So likewise it will go in a downward direction through this style and will reach in this ovary. The stigmatic surface provides the essential prerequisites for successful germination, which are absent in the pollen. This is very important point. Stigmatic surface provides the essential prerequisites, whatever is required for the development of the further steps. It is provided by the stigmatic surface to the pollen grain, because it is absent in the pollen. The pollen tube is finally pushed through the ovule and reaches the embryo sac. So likewise this pollen tube is pushed towards this ovule and it will go for this next step, that is called fertilization. The tip of the pollen tube enters in one of the synergids and then the products to release the content. So here you can see the tip of the pollen tube has been entering in towards the synergid sites, and it is going to be now release its content. Due to the pollen pistil interaction, what happens? Intense competition develops even in the compatible pollen grains gametes too. So because of this pollen pistil interaction, there is a intense competition uh, to be accepted on the stigma surface. So that's why for the process of fertilization in the sexual type of reproduction, pollen pistil interaction is very important process. It also plays important role in the sexual reproduction and the seed formation. Pollen grains can also be induced to germinate in a synthetic medium. Sucrose induces pollen germination and tube growth in vitro, in addition to the boric acids facilities and accelerates the pollen germination. It can be artificially also done by the artificial hybridization. It is one of the major approaches used in the crop improvement. All these are its significances and importance features. Only the desired pollen grains are hand pollinated and used for the fertilization. This hand pollinated means it is artificially done, and this is accomplished through the emasculation and the bagging and tagging procedure. About emasculation and bagging and tagging procedure, I'll explain you in the next lecture, which is very important and which is very interesting process done by the human. And this is about the pollen pistil interaction. As you know, pollen pistil interaction is very important, specific, in which pollen grains are accepted or rejected as per the need of the sporophytic tissue of the stigma, because it has that ability to accept or reject it. So it is very important for the process of the sexual reproduction. this is about this lecture, pollen pistil interaction. Thank you very much. Do not forget to subscribe my channel Patel Science Academy and be happy. Thank you very much.